Many RNAs inside our cells now we recognize as functioning in a non-coding way. The meaning of non-coding means they are not for producing proteins, but they're doing something else. And so we're starting to study what these RNA molecules are doing inside the cells and also inside the bodies. My lab has two major angles of study. One angle of study is to study non-coding RNAs in the blood cell system. And the particular type of non-coding RNA we're studying currently is the so-called glycosylated RNA. But it basically means the RNA is sugar-coated. Right, so it basically has sugar modification on the RNA molecules. And these sugar modification will enable the RNA molecule to do things that normal RNA molecules cannot do. So that's one aspect of it that's very interesting. The second aspect of it is almost all RNA inside our body are housed inside the cell. But these RNAs actually appear many times on the cell surface, on the outside cell surface of cells, which is again a very unusual place where they're supposed to be. So my lab was the first one to show a major function for this group of RNA. They actually control blood cells, how blood cells migrate inside the body in a significant way. And now we are really studying their relationship in disease settings, how they're controlling disease, and how we can potentially use them as a therapeutic target or be used somehow towards a therapy for human disease. The second area we're studying is what kind of information we can extract from the morphology aspects of tissues or cells. What is the shape of this cell telling us about this particular sample or this particular condition? We study neutrophils and looking at the shape of the cells versus what they can potentially tell us in terms of what they're doing. And it's amazing that there are quite some diseases. We know that these cells look different from other normal conditions, yet we have very little clue what those shapes are actually telling us. And now our research is actually starting to uncover this link between these abnormal cell shapes versus what they're potentially doing to the body. What my lab does is we try to use the best technology to address specific questions we have in mind. And in some cases, we have to develop our own technologies. One example is when we are studying the sugar-coated RNA, because it's a new field, the methodologies are quite limiting. So to do anything that's going to be interesting, we many times had to invent new techniques to do those things. And that's something also my lab is actually working on to develop those techniques. For the sugar-coated RNA, for example, one thing we're looking at is how these RNAs are dysregulated in disease settings, and especially in autoimmune diseases, because many of these cells we're studying is immune cells. We're also looking at the cancer relationship as well in that sense. The second aspect is to look at whether they can be used as a way, as a target for therapies. So we're developing new ways to perturb them or new ways to get rid of them or enhance them. In those ways, we're trying to develop new strategies for disease treatments.